Namaste. Good morning. Let me start sharing my screen. First, I would like to thank the Research Day organizers for inviting me to share our research with you today. Catholic University has been an amazing place for me to do research. It has been my home for more than 31 years. Today, I will share you my story, my journey with Bacteriophage T4, how we got into COVID vaccine design, and our experiences during this process and where we are and where we are going. First of all, what is Bacteriophage T4? Well, it is a virus that infects E. coli bacteria. It's a beautiful virus. It's a large virus. What you see is a reconstruction of a real specimen. Its head size is 120 by 86 nanometers, within which about 170,000 codes of genetic information is packaged. In fact, it is packaged so tightly that it generates an internal pressure that is about 4 times that present in a champagne bottle. It has a sophisticated injection machine, the tail and tail fibers. When it infects the bacterium E. coli, the DNA is injected into the cell. It triggers a series of reactions, literally tens of thousands of reactions that lead to synthesis or assembly of hundreds of infectious virus particles that burst open and are released into the medium and can now infect healthy cells. All this occurs through a precisely regulated program in a matter of about 20 to 25 minutes. In other words, Bacteriophage T4 is an incredibly fast and efficient killing machine or replicating machine. What got us into COVID vaccine design is our more than 3 decades of research at CUA. We were primarily interested in understanding the basic mechanisms that lead to the assembly of an infectious virus particle. We also harness some of that knowledge for biomedical applications such as vaccines and gene therapeutics. Here, I'm summarizing some of our 3 decades of research in 4 video clips that depict the mechanisms and atomic structures of Bacteriophage T4. First, how does bacteriophage infects E. coli bacterium? What you see are the structural models that are generated using a large amount of structural, genetic, and biochemical data. The virus lands on the bacterial cell. It recognizes certain unique molecules that are present on E. coli bacterium. And then the whole machine unravels. The tail literally drills a hole into the bacterial envelope, injecting its genetic material into the cell. How does the virus assemble? We have discovered a 12 subunit ring structure with dodecameric symmetry initiates the assembly of the whole capsid. This is the ring structure on which the whole capsid is built. It also has a central channel through which DNA passes during packaging into the capsid and released or exits the capsid during infection, and hence it is referred to as the portal. And this is present in almost all icosahedral viruses, and we have recently reported the first detailed structure of it about a year ago. What does the finished capsid look like? Here is a video clip that shows the entire atomic structure of bacteriophage T4 capsid. It is built with 930 molecules of a single protein subunit, but perhaps most important for today's discussion are these blue fibers that emanate from the capsid surface. These are known as Hoc molecules. Also, there are green subunits that are attached to the capsid. Those are called Soc subunit. Hoc and Soc are not essential for virus assembly or virus infectivity. Therefore, we can actually manipulate these molecules to incorporate foreign genetic material or foreign material into our virus. Finally, how does the virus package the genetic material inside the capsid? This is actually a fascinating story on which we spent most of our research. And we discovered that there is a nanoscale molecular machine that is assembled at the portal ring that I showed you earlier. And this machine comes with the genetic material, DNA, and using ATP as a fuel, it pumps the DNA into the capsid base pair by base pair until everything is packaged. It's a very fast machine. And in fact, T4 has the fastest packaging motor reported to date. All this mechanistic information was instrumental to engineer our virus to develop a novel vaccine delivery technology. And we use our virus as the backbone and splice genes into our virus so that they could mimic the pathogenic, whichever the genetic material we incorporate in it. For instance, if we want to make a COVID vaccine, we would splice genes corresponding to the COVID viral genetic material, and some of it displayed on the virus capsid mimics the COVID virus. And when exposed, our immune system would generate responses that can protect us against COVID infection. This is a platform technology, which means we can incorporate any pathogenic material depending on which vaccine we would like to create. We can create a flu vaccine if we would like to incorporate, if we splice the genes corresponding to the flu virus. And in fact, recently we have reported a dual anthrax plague vaccine that can protect against both these deadly biothreat agents. Coupled to this, in parallel, we have recently developed a powerful CRISPR genome editing technology that allowed us to engineer our virus and generate these chimeric viruses in an accelerated fashion. So we have been well prepared to take on COVID vaccine design. But what motivated us the most was the distress and suffering Wuhan coronavirus was causing in China. Wuhan has a special heart in my place, in my, sorry, special, is a special place in my heart because many of my postdocs, graduate students, came from Wuhan and contributed tremendously to our research program. And also considering the reports coming out from China, as a virologist, it became clear to me that this is a dangerous and highly transmissible virus, and it is only a matter of time that we might face such a predicament in the United States. So it was very upsetting. And I gathered my team and discussed the possibility whether we can suspend our research projects, the ongoing projects, and direct our resources and efforts towards COVID vaccine design and contribute to the emerging pandemic. This was going on in the end of January, beginning of February, but we have nothing to start with, no COVID genetic material. So I wrote to Dr. Tony Fauci on February 18th. I know Fauci from my previous interactions with him during the development of HIV vaccine. And I will share a couple of sentences from a fairly long email in which I asked his advice as well as requested some genetic materials from NIH. Considering the emergency situation and the enormous suffering the Wuhan virus is causing, we cannot sit by and not do our part. Therefore, with minimal resources available to us, we have decided to go ahead and construct vaccine candidates which will be ready for testing in about 4 weeks. So the key point here is we want to contribute. We didn't know where we would go with it, but we couldn't help but really do something about it. At the same time, I have also written to several people, among whom are Barney Graham and Kizzy Corbett. These are the two key architects of the current Moderna vaccine. And I know they are from VRC, Vaccine Research Center from NIH, which has the COVID genetic material. So they kindly agreed to supply the genetic material, although it took a little bit of time to go back and forth and the paperwork. Finally, on March 9th, 2020, we received the genetic material from VRC NIH. At the same time, we are also planning the COVID design. And this is probably the first detailed blueprint of T4 COVID vaccine on a piece of paper, but it is quite detailed and very complex, actually. And you see many arrows and these red flags here. These represent alternatives and backups because we did not know which design will be effective. And we wanted to cover all the bases and use an iterative process to design our vaccine and test them. And in fact, in the end, we have designed many more constructs than we have originally planned because of the some of the challenges that we faced along the way. But we did have a nice blueprint to start with. So intense work had begun, and we restricted our space in the third floor biology building, and then came the lockdown. And in fact, there was a possibility of shutdown. Luke Allard, our safety director, called me one afternoon. He knew that we have been working very hard began our work and working very hard towards the design, COVID vaccine design. He called me one afternoon and said, look, there might be actually a shutdown, meaning that we will not be able to go back home. We will have to stay in the campus, and he would provide arrangements for accommodation and food, etc., for us to stay. It was a pretty tough time, pretty scary time. And again, the team went back and forth for an hour or two. And finally, we decided no matter what, we will stick to our plan and keep working. Fortunately, the shutdown never happened. Still, it was not easy. It was pretty tough, especially people who are working in the lab. I will mention that at the end of the talk. And they worked 12 to 14 hours a day, 7 days a week, very hard, pushing as hard as possible to design and contribute to the pandemic. And I'm going to take a few minutes to discuss the construction of the T4 COVID vaccine and some unique features. Unlike the Moderna, Pfizer, and other vaccines, which focus on a single component of the SARS-CoV-2 virus, which is the spike protein, our vaccine, while we also focus on spike protein because it's a critical target, but we also incorporate additional viral structural components. The nucleocapsid protein, envelope protein, and membrane protein. Our prediction and expectation is that we eventually need broader and more robust protection against this coronavirus and potential variants and also probably emerging coronaviruses. So we wanted to incorporate other viral structural components that are actually conserved in most of the coronaviruses. Second, our vaccine formulation would be adjuvant-free, meaning no strong chemicals would be used in our formulation. These chemicals are generally used in almost all the vaccines in order to stimulate immune responses. We constructed this by starting with SARS-CoV-2 genetic material. Using structural modeling and bioinformatics, we identify the genetic sequences corresponding to spike, nucleocapsid protein, and other components, and inserted those genes into recombinant plasmids by constructing these recombinant plasmids and showing these different colors representing the COVID genes. We then transformed these plasmids into E. coli, and then infected these recombinant E. coli with our bacteriophage T4. When it injects and replicates, the virus exchanges, there are genetic exchanges between its genome and the plasmids that are residing in the bacteria, generating the chimeric viruses, now carrying pieces of coronavirus genetic material. In order to identify the sites where we can insert these genetic or exchange these genetic sequences, this is a technical side. I will not have time to go through that, but just to make the point, the circle here represents the viral T4 phage genetic genome with all the essential genes for head, tail, replication, and so forth. But we found there are 4 sites represented by arrows where we can insert COVID genes without disrupting the essential functions of our virus. That's important because we want to produce the chimeric virus that is functional and would be able to display the COVID genes. So by iterative infections and CRISPR genome editing that I mentioned to you before, we could create many combinations of these COVID genes incorporated into our chimeric viruses. And those are represented by different colors here. And these chimeric viruses are vaccine candidates. We have literally generated hundreds of chimeric viruses with various combinations of the COVID genes, of which we selected 12 of them for testing in mice. And analyzing the responses that the mouse generated, we found that one of these candidates gave the best immunogenicity and protection. And let me share with you a video clip that depicts the components of our T4 COVID vaccine. This is our T4 phage. It has no hock and salk to start with. We incorporate COVID genes, like you see in colors. The rest of it is in gray. That is the phage genome, its own genome. In addition to that, the vaccine has these blobs that you see, hopefully, on your screen. These are nucleocapsid protein molecules that are packaged inside the capsid. We also decorate the capsid by fusing the envelope peptide to the Hock fibers now displayed on the capsid surface. In addition, we also decorate the capsid with trimers, the spike trimers that are produced in Chinese hamster ovary cells through Salk molecules. As a result, our COVID vaccine has COVID genes, nucleocapsid proteins, envelope peptides, Salk primers that are all incorporated in a single nanoparticle structure. That is our vaccine candidate. Or that is the one that gave the best immunogenicity and protective efficacy. Now, how effective is it? Next few slides, I will go over some of the pieces of the data that speak of the vaccine efficacy. So it is very effective. This vaccine, we have two versions of it. It's not important. They are very similar versions of the same vaccine. They produce, we basically immunize or vaccinate mice and take the vaccinated blood and collect sera from it and analyze for various immunological assays. And this particular histogram shows high titers, strong spike-specific antibody titers greater than 1 million. Of course, the control is just the phage. It has no COVID vaccine, COVID virus components, shows no significant antibodies, and we use two methods to analyze that. Yet these antibodies also block virus. This is SARS-CoV-2 attachment to its receptor. There is a biochemical assay here, cell culture assay, where you see fluorescence. Green fluorescence means virus can attach and enter. And in the presence of our vaccinated sera, there is no fluorescence, but that means virus cannot attach to the receptor, meaning it cannot enter the human cells. Strong virus receptor blocking antibodies. And most importantly, perhaps, I'm sure you all heard about virus neutralization titers in the news. It produces strong virus neutralization titers, up to 5,000, that would lead to 100% virus neutralization in these assays. Finally, unlike the other vaccines, our vaccine produces strong nucleocapsid and E-specific antibody titers as shown here. So therefore, it provides or it generates broader immune responses when compared to the other vaccines that are focused only on the spike. In parallel, in collaboration with Virovax Inc., headed by Dr. Sunil David in Kansas, we conducted a rabbit study where the rabbits were immunized with our best vaccine, the two versions of it, and again, looked for these responses in the sera collected from the blood from these rabbits. The vaccinated rabbits produce, again, high titers of spike-specific antibodies, and again, very high titers of virus neutralization antibodies. In fact, the rabbit titers are about fourfold higher than what we obtained in the in mice sera. Now, the question then is, do these antibodies protect the mice against COVID infection? When we started the work, there is really very little known about this virus, certainly no challenge model. By October, when we finished all the mouse studies, a mouse challenge model has been developed by researchers at University of Texas Medical Branch, UTMB, Galveston National Laboratory. This requires biosafety level 3, which obviously we cannot do here. So I contacted Ashok Chopra, a colleague of my, collaborator of mine for more than 10 years for the anthrax and plague vaccine. And we are all very much interested to test our mice, whether they can be protected against a lethal challenge with the SARS-CoV-2 virus that UTMB has developed by Ashok Chopra's colleagues. So the question is, we have to ship our mice to Texas. And that was, we went through major trials and tribulations for that. It was a logistical nightmare, as many. I would love to share that story with you, but I don't have time today. But suffice it to say that we finally, on November 2020, we managed to put our mice on flight to Houston, Texas, and then onwards to UTMB Galveston. It was a very, very nervous time for all of us, the team, especially for some of us who actually worked with the mice every single day. And both for the mice, safety of the mice, as well as all the investment we made in designing the vaccine and testing and so forth. So we were relieved when it was clear that finally the mice were housed in the biosafety level 3 facility at UTMB. Very grateful for that to have happened. So this is all has been worth it, because the challenge showed that the COVID vaccine that we designed is protective, and in fact, it completely protected mice against lethal SARS-CoV-2 challenge. Let me share some pieces of data. Here is a graph showing with the days after challenge. Basically, your mice are challenged with a highly lethal dose of the coronavirus developed in UTMB, and every day it was followed. The weight of the mice were followed, and the weight decreased rapidly. Within 5 days, there is a loss of 20 to 25% of weight in the control animals, meaning animals that are treated only with the phage but with no COVID vaccine. And with the vaccinated mice, again, we have two groups with two versions of the same vaccine. There is a slight dip in the initial couple of days and quickly regained, and none of the mice actually significantly lost the weight when compared to the control mice. And in terms of survival, 80% of the control mice, in fact, when they lose 20-25% of the body weight, it's very, very significant, very bad. The mice basically collapse at that point. 80% of the control mice died at 5 days after challenge, and the rest slowly, eventually regained the weight. But 100% of the vaccinated mice, they all survived. So combined with the immunological data, this is a clear demonstration that the T4 COVID vaccine is effective. It has both immunogenicity and the productive efficacy. Now, where are we now and where we are going with this vaccine? As I mentioned to you, we have an effective T4 COVID vaccine. These are the two little vials of the vaccine that reflect the vaccine I just talked about. As I already pointed out, our vaccine has broader and more robust, or expected at least more robust protection, because it has conserved viral structural components that are not present in most of these vaccines. In fact, any of the vaccines currently available. So it is expected that it would generate broader protection against the variants and also future emerging coronaviruses. It is something we are actually testing as we speak in another study at UTMB. And we expect the data to be out in about a couple of weeks. As I also pointed out, our vaccine is adjuvant-free. There are no chemicals, adjuvants that are part of most of the vaccines, in fact, Moderna and Pfizer, and also no infectious viral material. The adenovirus-based vaccines in J&J and AstraZeneca. And stability-wise, we expect our vaccine to be very stable. We are conducting those studies as well, because phage as a backbone is a very stable material. In fact, they survive in very hostile environment. So our vaccine certainly doesn't require freezing. In fact, freezing would be detrimental. It might even be stable at room temperature. We are also producing the vaccine. Adaptophage Therapeutics, Inc., a Gettysburg company, has GMP manufactured our vaccine and actually did an independent study and confirmed the results, some of our results. And we are moving towards a hamster study before we can transition this to phase 1 human clinical trials. At the same time, we are at the final stages of finishing up an FDA IND application so that all this hopefully will come together by summer so that we can conduct a phase 1 human clinical trial. And this effort has been funded by DHA, Defense Health Agency, which is part of the DoD. What are the potential applications of the T4 COVID vaccine? First, it could be used as a booster vaccine. This pandemic is by no means over. It is still evolving, and very likely all of us who have been vaccinated likely would require 1 or more boosters before the pandemic is controlled. It is probably better to have a booster vaccine with a broader and more durable protection than one that is entirely focused on the spike protein, because you can't keep on giving different versions of the spike vaccine, and that may not be practical. Keep in mind that the current vaccines are emergency use authorization vaccines. They are not FDA approved. FDA approval comes for a second generation vaccine, whatever that might be, or whichever those might be. Those will have broader and more durable protection and should have very high safety profile. So still there is a lot of time to determine those, and we hope that T4 COVID vaccine would be one of those. And most of the global population is still unvaccinated. Current platforms unlikely would meet the need of complete global vaccination, at least a great percentage of global vaccination that is eventually required to control this pandemic. So platforms such as ours, which can be relatively easily produced and can be distributed, would be very useful for these efforts. And we do hope that this is an optimistic projection. We do hope that our vaccine will come on board at the beginning of 2022. So what's beyond the COVID vaccine? As I mentioned, it's a platform technology. It could be applied for designing vaccines against any pathogen. And in fact, we have already begun work on a universal flu vaccine, which we would like to see that an effective universal flu vaccine that would perhaps obviate the need to vaccinate against flu every year. That is the current platform and the current model. So hopefully a universal vaccine would not require such a yearly vaccination. We are also very excited about a possibility to construct a dual flu COVID vaccine. This will be challenging, but something we are very excited about, and we have already begun developing a blueprint for that. And beyond that, other vaccines are also under consideration, such as Zika and Ebola and vaccines, which is great, needed. But beyond the vaccines, in fact, before COVID appeared, we were actually going full force with gene therapy experiments and developing some gene therapeutics for stem cell therapy, cancer therapy, and so forth. And we would like to get back to that and continue that work. And we have begun to work on the sickle cell disease and muscular dystrophy disease gene therapy application of our phage platform. Of course, all of this would require enormous resources and infrastructure and a very strong research, cutting edge research program. And this is one of the main reasons why we have recently established this Bacteriophage Medical Research Center, because we could use it as a vehicle to raise the necessary resources and develop the programs for training the students of the cutting edge research, as well as benefit the CUA community at large, and use the center as a vehicle to raise resources, both from NIH and the other agencies, but also from private individuals and foundations, and at the same time, establish and continue establishing collaborations with biotechnology companies so that our research results could be transitioned to products that could be useful for healthcare and other biomedical applications. So I would stop here and would like to take a couple of minutes actually to express my deepest gratitude to numerous students and postdocs who worked with me over the past 3 decades. They made great contributions, especially today. I would like to thank few people who worked very hard for this COVID vaccine. They are Dr. Jingyan Zhu, and here worked tremendously hard. Niti Ananthaswamy, both are research fellows, postdoctoral research fellows. Wai-Chun Tang, Swati Jain, and Himanshu Batra. They are graduate students actually, but after they finished the PhD, they decided to stay back and really help us with the project. And that was very nice of them to do that, because a lot of it involves not lab work, but also animal work and so forth. Miss Isabel Nunes, who was an undergraduate student, who also, after she finished her undergraduate program, really volunteered to offer her help to stay and work and help us with the animal studies and took care of the animals beautifully. And Mr. Ralph Albano, our vice provost, who has been a tremendous support over the 3 decades or many, many years, and who was instrumental in establishing the center I just mentioned. And a special thanks to Ms. Mera Camden and facilities personnel. They have been a tremendous help. Everything that you can think about that could break down did break down during this lockdown. They were there, right there to help us. And in fact, there was a time the animal facility AC was broke down, and the temperature came to 65 degrees one morning when Swati went and checked the temperature, and we were all very worried, and the mice they all gathered in one corner and kind of sat or laid down on one top of each other to feel warm. We had really a tough time to deal with that. Facilities were right there. In fact, we brought our space heaters and in fact, hair dryers. We used the hair dryers to sort of warm up the cages to keep the mice reasonably comfortable, because they're very, very sensitive to cold actually. This is just before we were shipping the animals to UTMB. So finally we managed that, and with a sigh of relief, and facilities were tremendously helpful, and I couldn't thank them enough for that. My colleagues, Ashok Chopra, and others, and his colleagues at the UTMB Galveston National Laboratory, and Virovax, Dr. Sunil David, who conducted the rabbit study, and Adaptophage Therapeutics, Dr. Suben Basu, who is the COO, and his colleagues. And finally, NIH and National Science Foundation, which both the agencies provided the funding for the last about 25 years or so. We have been funded by both NIH and NSF. We are very grateful for their support. And finally, our little guy, Bacteriophage T4, the models you see the students are holding, which is I can say it is the love of my life. Finally, thank you all for listening. I'd be happy to take any questions.
Thank you very much, Dr. Rao, for your great talk. I personally learned a lot about this bacteriophage and your vaccine development. The story, like we have to ask you, like how you might ship to Texas. The audience has many questions, and then throughout the time, I guess that's the beauty of the webinar. People have questions, and you already addressed many of them. But there's still some questions, and I'd like to ask you. The first one would be, thank you, Dr. Rao. Your research is life-saving. My question is, what role researchers now playing in combating vaccine disinformation, and how can audience help with this effort?
Yeah, that's a big problem. Scientists and policymakers have been working very hard to provide the science and information and disseminate the public in the best possible way we can. And the main way I can think about as a scientist is like this: a communication. This is a research day. Of course, it's specific to Catholic University, but there would be other venues where more public might be interested in, and we should communicate the data as well as address our concerns, because for them the concerns are very real, and there are some issues with the vaccines as well. So we need to really communicate so that we trust each other, as well as clarify some of these issues that are concerning to the public and so forth. So that's the best way.
Thank you very much. The next question is, I guess you addressed as well at the last slide, but the audience probably would like to hear again, elaborate a little bit. What role are the Catholic University students playing in your research and the development of the vaccine?
Yeah, Catholic students are the engine, basically. They run the research machine. I have, over the years, undergraduates, many undergraduate students, even actually high school students coming from local area high schools, and many graduate students and postdoctoral research fellows. So that's probably the most consequential, most joyful aspect of my career as a scientist, to work with the students and postdocs and so forth, and see them shape their career and actually do great things in their own careers now, many of them. So that's core fundamental aspect of at least my research program at the Catholic University.
Thank you for addressing that. The next question, people asking, because you already talked about this technology can be applied for different biomedical applications. So the specific question is, like, could the bacteriophage be used to generate an effective vaccine for lung disease?
Lung disease. Yeah, lung disease, I would think it would be more of a gene therapy type of thing, depending on which disease it is. If there is a defective gene that is causing the lung disease, or some immune system that is autoimmune antibodies or something that's attacking the lungs and so forth, or inflammation, those sorts of things. So some of them are very complex and we may not have enough information. But if there is a particular genetic defect that can be cured or treated by providing a good gene, then I think our system would be potentially be useful for that. And I think that's why we are working with the sickle cell and muscular dystrophy, because they're very well understood, they're very focused, and we could actually make that as a starting point, and eventually it could be applied for more complex tissues like lungs and other tissues.
Thank you. There's another question from our audience, is like, is there any interesting research on hold that you are excited to go back after COVID pandemic?
I'm sorry, cold, you said?
Other research that you kind of step into the COVID-19 vaccine development. Is there other research that exciting you're working on you would like to continue as well?
Yeah, so I didn't talk about it today. I just mentioned the initially some basic research, and I should mention our basic research is also the engine that is important for biomedical applications and so forth. So I would, if I have my fantasies come true, I would like to see the whole packaging machine at the atomic scale in a dynamic state of packaging DNA. And we are actually trying to do that as deeply as we possibly can using single molecule biophysics, structural biology, and all that. I didn't have a chance to talk about it. There are many papers were published, but I would, of course, love to continue that and love to see that happen at some time in the future.
Thank you very much. Yeah, there's a lot going on, I know, in your research lab. There's another question is like, thank you, Dr. Rao, for the nice talk. I would like to know how much effective would be the vaccine against COVID variants. If yes, how will it be work against variants?
Yeah, so that's a very important question, because right now the variants is a big problem, as you all know. As I mentioned, we are conducting a third mouse study in UTMB, Texas, and that is the very question that we are addressing actually, as well as more broadly, whether our vaccine would be effective against the variants. My prediction is that, in fact, in the vaccine production that we are going to make, we have introduced the variant mutations into our vaccine so that it would be even more broader and robust. But my prediction would be, yes, it would be, but we need to wait for the data.
Thank you very much. It's really a fantastic talk. It's learn about the difference of your vaccine from others, and so many things, mechanisms, and then you give the way that the general audience, I hope, get this take of what you have been working on, exciting research happening on this campus. But for the sake of time, we would like to wrap up this session now. If the audience have more questions and you want further discussion, please contact Dr. Rao offline. Thank you again, Dr. Rao.
Thank you, Shaolong, very much, and thank the organizers. Once again, I would express my deep gratitude to all the students and my fellows and the Catholic University for supporting us. And I wish you all a wonderful rest of the Research Day program. Thank you.